Okay, these are the plant parts in the models. So this is obviously the flower. The parts you need to know for the flower are the petal, which is the pink part here. Obviously it's used to attract pollinators for the most part. You also need to know the sepals. So these are these little green looking sort of like leaves, but they're actually cover the petals when the flower is in a bud. Those are the sepals. Then we have, we'll start at the top. So we have up here the stigma. This whole thing is the female reproductive part. So this is a complete flower. It has both male and female parts. So the stigma is the sticky part where the pollen lands. Then we have the style, which is this long part here. You can see part of it in this picture as well. So that's the part where the pollen tube grows down to reach the ovary. So the ovary is down here. This is not a great model. There's a cutout model or a section model that's better, but the ovary is going to be inside this area. And so the combination of the stigma, the style, and the ovary together is called the carpal. You can also call it the pistil. We'll usually call it the carpal. And that's the female reproductive structure. Then we have the male reproductive structure. So there's these parts here where the pollen's produced. Those are the anthers. Then we have the white parts here. See them here? Those are called filaments. And the anther plus the filament altogether is the stamen. You can find all these parts in your book as well as your lab atlas and your symbiosis lab manual. So that's the flower. Here's a root. There's only 3 parts of the root. This is the cortex. It's the bulk of the root. This is the vascular cylinder. It's where the xylem and phloem are located. And these are the root hairs, which are labeled C here, but those are the root hairs that increase the surface area for absorption of water. So this is a dicot root specifically. Monocot roots look a little different, but those are the parts you need to know for this. This is the leaf. The leaf will start at the top. So there's this part here. It's called the cuticle. It's also on the bottom of the leaf. So it's the covering that prevents water loss, waxy covering. Then we have B, this layer of cells at the top and the layer of cells at the bottom. So that's called the epidermis, upper and lower. We have these cells, which are labeled C here. So that's called the palisade parenchyma. These cells that have lots of airspace in them, those are called the spongy parenchyma. And both of the C and the D together, both the parenchymas together, is the mesophyll. E is pointing to a chloroplast, so all the little oval green structures are chloroplasts. Down on the bottom side of the leaf, the lower half of the leaf, we have these openings called stomata. So here's one that's a cutaway. Here's another one. Stoma is singular of that. It's where the gases are exchanged. So carbon dioxide comes in, oxygen goes out, water can also go out. Guard cells are these cells that are on the side of the stoma. They fill and shrink with water as well as ion movement and microtubule movement or cytoskeletal movement. And when the plant is releasing less water, when it's dry out, they will swell and close the stoma. When it's not as dry and there's more oxygen carbon dioxide exchange, they will shrink and they'll open the stoma. This is the vascular bundle. It's like the vein in the leaf. This is the vascular sheath, the yellow part. And then we have our xylem and our phloem. So the xylem is in the red up here, and the phloem is in the blue. This is a dicot leaf, you can tell because it has the palisade and the spongy types of mesophylls. This is a stem, so it's a relatively young stem. It's a dicot stem. So the parts here are the pith in the middle. Then we have the xylem, the primary xylem in golden. We have the vascular cambium in this little yellow line here. It's where the new cells are produced. It's the stem cell producing part, the stem cell containing part of the stem. So it's going to make new xylem on this inside and make new phloem on the outside. And that's what these chunks of red are, or the red pink. Those are phloem. That's phloem. You can see all the phloem here, channels carrying the food. And then we have this outer layer here. You can see it in the light tan. That's the epidermis. And the very outer side is the cuticle. So that's this dark part here. This is specifically a lime tree. Tilia is the species, T-I-L-I-A. It's a 1-year-old stem. Here's an older stem, and we start in the middle again. Again, this is just like a pie piece from the trunk. So the pith is in the middle, this A. Then B is going to be your primary xylem. So this is the first layer of xylem that was put down. You can see that ends here. Then C, this chunk here, is the secondary xylem. So that's the second layer of xylem that was put down. And that's going to be this region here. The place where the xylem and phloem are made is D. That's the vascular cambium. The stem cells are located there. They divide, and the xylem is in, the phloem's out. So you can see why this is secondary xylem and primary, because as the vascular cambium puts down more cells, they're going to be further and further out. And so these are the rings of the tree trunk, the different layers of xylem. E here is the secondary phloem. Primary phloem is already shed off the tree, or it's incorporated into the bark. So this is the secondary phloem in E. It's really these types of cells here, all these pink ones. And then F, this whole chunk, including the brown as well as this green. So this part here, that's the bark. So the bark's not just the brown on the outside, the bark actually includes the remnants of the phloem from the year before. And then G itself is actually cork. And so that's usually what we really see as what we think of as bark, is the cork part there. So those are the parts of the models that you need to know for the plant.